The search for ways to harness the power of the immune system in the fight against cancer has led to the development of chimeric antigen receptors, or CARs. CAR-T is not a pill, chemotherapy or transplant. It is a new type of therapy, known as a CAR-T therapy, that uses the power of your own immune system to treat your cancer. It can be used to treat diffuse large B-cell lymphoma, or DLBCL, in adults, and B-cell acute lymphoblastic leukemia, or ALL, in children and young adults up to 25 years of age. To understand how CAR-T therapy works, we need to first look at what a CAR is. A CAR is a special type of protein that scientists can design to recognise certain cells in your body. This CAR protein is attached to your T cells, which is why it is called CAR-T cell therapy. This treatment is made of CAR proteins that are attached to your own T cells. Each CAR protein contains 4 parts, which play different roles in finding and destroying your cancer cells. The part of the CAR protein that sits on the outside of the T cell recognises and attaches to the cancer cells. The other parts of the CAR protein tell your T cell to destroy the cancer cell. CAR-T only recognises B cells because it has been designed to recognise a very specific protein called CD19, which is only on the surface of B cells, or cells that will eventually become B cells. This is important because it is these B cells that we want to recognise and destroy. After being identified as a candidate for CAR-T therapy, you will have your T cells and other white blood cells collected from your blood by a process called apheresis. During apheresis, only your white blood cells, which contain T cells, are removed, and the remainder of your blood is returned to you. Your cells are then frozen and shipped to the manufacturing facility, where they are reprogrammed into CAR-T cells. Once your cells arrive at the manufacturing facility, they are thawed and then washed, and special steps are taken to get your T cells ready to be reprogrammed. The genetic reprogramming of your cells is done using an inactivated virus, which brings the gene for the CAR protein into your T cells. It is these genetic modifications that reprogram your T cells into CAR-T cells. Once there are enough of the CAR-T cells for treatment, they are frozen and shipped back to your treatment centre. Before you receive your CAR-T cells, you will be given lymphodepleting chemotherapy to prepare your body for the CAR-T cells. When your reprogrammed T cells or CAR-T cells are infused back into you, they start looking for the cancer cells. When a CAR-T cell finds a cancer cell, it attaches to it. When this happens, a series of events occur that tell the T cell to destroy the cancer cell that it is attached to. When this happens, your body may produce other CAR-T cells, which can also recognise and destroy the cancerous B cells. Once the initial wave of cancer cells have been eliminated, some of the CAR-T cells will remain in your body to keep a lookout for any new cancer cells.